Buffers. Buffer is a chemical mixture which resists change in pH either by the addition of acid or base. This means that buffer is a mixture of chemicals, and these mixture of chemicals takes care of fluctuation in pH, means it does not allow the pH to change. The pH of the system, it does not change due to this chemical mixture, and pH may be changed due to addition of acids or bases. So buffer, it is a solution which does not allow the pH to change, which may be due to addition of acid or base. This addition of acid or base should be in less quantity. Therefore, the more improved definition of buffer would be: buffer is a chemical mixture which resists in pH either by the addition of small amount of acid or small amount of base. That is should be the correct and proper definition of buffer. The another definition is these are aqueous systems, which is an aqueous solution, a solution prepared in water with different chemicals that tends to resist changes in pH when small amounts of weak acids or bases are added. So weak acids are represented as hydrogen ion concentration. That means we have learned what is the definition of acid. These are the proton donors. That's why it is shown here, the hydrogen ions. And we have already learned what are bases are proton acceptors. So therefore it is mentioned as hydroxyl ion concentrations. So either by the addition of H plus ions or OH minus ions. If these are added into that system, there is no fluctuation in pH. That is called as buffer. We always say that buffer is a system, means it is a combination of chemicals. That is why it is called as a system that take care of acids, a system that takes care of bases. So this buffer system, it is a chemical mixture. What are these chemicals? These are weak acid and its conjugate base. So buffer is a mixture of these two, weak acid and its conjugate base. We have already learned weak acid is proton donor and conjugate base proton acceptor. That means in a buffer system, it is a combination of proton donor and proton acceptor. So if these two are present in the system, proton donor can take care of bases, proton acceptor can take care of acids. Say for example, a mixture of acetic acid. Acetic acid is a weak acid. Mixture of acetic acid and its salt, such as sodium acetate, that is acetate ion. So acetic acid is a weak acid and sodium acetate is conjugate base. So when these two are mixed, it leads to formation of acetate buffer and it forms a buffer system. Almost every biological process is pH dependent. Cells, organisms maintain a specific and constant pH in cytoplasm because if we consider cytoplasm, it is a mixture of several things. Several metabolic processes are going on, several biochemical reactions are going on, which leads to production of acids and bases. Still, the pH, total pH of the cytoplasm does not fluctuate. Why? Because there is presence of a buffer system in cytoplasm. So this keeps the biomolecules in optimal ionic state, usually near the pH of 7. So buffer systems are required everywhere in animals body, in human body, in cells, fungal cell, bacterial cell, protozoal cell, etc., etc., because the biochemical reactions are dependent on pH environment. In a multicellular organism, the pH of the extracellular fluid, like in our case, it is blood, is tightly regulated. The pH is maintained by biological buffers, mixtures of weak acids, and their conjugate base. That means in every living system, buffers are naturally present, which does not allow the fluctuation in pH. For example, in case of animals, if the pH of the blood it fluctuates, then it may lead to hazardous effects on the life of that particular animal. Most of the microorganisms they tend to grow at pH values between 66.5 to 7.5, means they are the most suitable environment is near neutral, and few of the microbes they grow below pH 5 or they may grow above pH 9. These organisms are extremophiles, acidophiles. Many microorganisms produce acids or alkaline substances that can alter the pH of surrounding environment, that may be unfavorable for the growth of other microbes or for themselves. Therefore, when the microorganisms are grown in laboratory, buffers are added to the microbiological media to maintain the desirable pH. See this equation very carefully. This equation is called as Henderson-Hasselbalch equation. This equation is very important in buffer chemistry. Let us see what is this equation. This is pH equals to pKa plus log of proton acceptor concentration divided by concentration of proton donor. This equation is very important for the calculation purpose, that is Henderson-Hasselbalch equation. So this equation gives the quantitative relationship between pH, the buffering action of the mixture of acid and its conjugate base, and pKa of weak acid. So this quantitative relationship is given by this Henderson-Hasselbalch equation, which is stated in this equation. So this equation defines the quantitative relationship between pH, buffering action of weak acid and its conjugate base, and pK of weak acid as stated above. This equation is of great importance in buffer chemistry because it can be used to calculate the pH of the solution. This part is very important. It can be useful for the calculations of pH of the solution if we know the molar ratio of buffer ions, that is conjugate base, that is proton acceptor divided by proton donor, as stated in earlier equation. So if this ratio is known, then the pH of the solution it can be calculated, and if the pK of weak acid is known. So if we know these two, this one and this one, we can calculate the pH of solution. Also, if the molar ratio of weak acid and its conjugate base, that means this proton acceptor divided by proton donor, this ratio, uh, which is necessary to prepare a buffer solution. So if you are interested in preparation of a buffer solution, you need to add these two chemicals. So what should be the concentration of this and what should be the concentration of this? It can be calculated for specific pH if the pKa is known. So this is how, if you can calculate this molar concentration, or you can calculate the pH. So all these things are relevant to the preparation of solutions, buffer solutions having different strengths, having different pH. Now, a solution containing both HA and A. So what is this solution containing HA and A? It is a buffer solution. Okay. So this buffer solution has capacity to resist change in pH. Means in this particular solution, if there is addition of small amount of acid or small amount of base, the pH of this system does not change. That is why it is called as, it resists the fluctuation in pH. So that is the function of buffer. See a condition in which if acid were added to buffer solution, this is the buffer solution. If there is addition of acid, what is acid? Acid, there is addition of H plus ions. So if these H plus ions are added to this buffer solution, what will happen? These are added to the solution. So we know that buffer system, it contains these two. Out of these two, this A minus will go and it will react with this acid, that is proton donor, and immediately it will convert it into HA. This is how effect of free H plus ions is neutralized. That means there is no free H plus ions. These free H plus ions are neutralized by this conjugate base to form HA. Similarly, if in this system base is added means it will be hydroxyl ions, proton acceptors. So among these two, this HA will react with OH minus. That means HA, weak acid, will react with this base to form A minus, that is conjugate base, plus H2O. See the magic of the system, that this is the buffer which contains these two chemicals, weak acid, conjugate base. What do we see in this diagram? That there is formation of HA, there is formation of A minus. This means that even there is addition of acid or base, these acids and bases are neutralized to form weak acids and conjugate base. Means same type of chemicals are keep on synthesizing, so that there will be no formation of free H plus ions or free OH minus ions. That is the function of buffer system. Buffer system does not allow H plus ions to be free, OH minus ions to be free. If H plus ions are free, the pH becomes acidic. If OH minus ions are free, the pH becomes alkaline. But it is avoided by means of A minus, that is conjugate base, and HA, that is weak acid. This is how the buffer system works. Now let us see few practical examples. The buffers that we prepare or use in microbiology laboratory are phosphate buffers. This phosphate buffer is prepared by using a mixture of these two chemicals. Buffer is a mixture of chemicals. So these two chemicals, one is proton acceptor and another one is proton donor. So in this case, in phosphate buffer system, disodium hydrogen phosphate, it is proton acceptor, and sodium dihydrogen phosphate, proton donor, weak. So both are the salts. So these two will form a system, and that system will take care of acids and bases. Let us see how. So there is a mixture of this two. So this mixture will take care of, it will avoid the change in pH due to addition of either HCl or NaOH. Say for example, if there is addition of HCl, what will happen? This HCl will react with one of these two. In earlier slide we have seen that when acids are added, acids will react with conjugate base. So here in this system, the conjugate base is this disodium hydrogen phosphate. So this disodium hydrogen phosphate will react with HCl to form NaH2PO4, that is sodium dihydrogen phosphate plus NaCl. So what is the sodium dihydrogen phosphate? It is a component of that buffer system. For example, if there is addition of NaOH to this system, what will happen? This, as we have seen in earlier slide, if the bases are added, these bases are neutralized by weak acids. In this system, these bases will be neutralized by this weak acid, that is sodium dihydrogen phosphate. So this sodium dihydrogen phosphate will react with this NaOH to form Na2HPO4, that is disodium hydrogen phosphate plus water. What it means? It means that we can clearly see, even if there is addition of acid or a base, there is no formation of free H plus ions or free OH minus ions. If there are no formation of free H plus or OH minus, it means that pH is going to remain stable. We can see here, even if there is addition of this acid, it leads to formation of weak acid. Even if there is addition of base, it leads to formation of weak base. So same type of compounds which are the constituents of buffer are reformed even after the addition of acids and bases. Another example is of acetate buffer. Acetate buffer is prepared by using weak acid, that is acetic acid, plus its salt, that is sodium acetate, CH3COONA. So it forms a buffer system. Now let us see how this buffer system actually acts. So whenever there is addition of acid like HCl, so this HCl will react with conjugate base, that is CH3COONA. So CH3COONA plus HCl, it will produce weak acid plus NaCl. If there is addition of NaOH to this system, this NaOH will react with weak acid and it will form CH3CONA, that is sodium acetate plus H2O. So we can see that even there is addition of acid or base, similar chemicals are produced which forms the buffer system. So that is the beauty of this buffer system. तेच ते केमिकल्स परत परत तयार होत राहतात की ज्याच्यापासून हा बफर बनलेला आहे. Now, what is the most effective buffering system? So most effective buffering system contains equal concentration of HA and conjugate base. That means equal number of proton acceptors and equal equal number of weak acids proton donors and equal number of proton acceptors. So if the concentrations of both are same in a buffer system, that will be called as an effective buffering system. According to Henderson-Hasselbalch equation, when concentration of conjugate base means proton acceptors equals the proton donor, that is weak acid, then pH equals to pKa. Therefore, the pK of weak acid base system represents the center of buffering region. The effective range of a buffer system is generally 2 pH units centered at pKa value. So the effective pH range for a buffer is equal to pKa plus minus 1. So this is the effective range for a buffer. Effective range means it is more capable, more effective in neutralizing acids and bases.